All right.
There's a big group. So there's some actually that are invisible on the walls as well. So you're seeing some of us.
They're hiding. Okay. Well, hi, everyone. I'm really glad you invited me. I'm always excited to talk about this work, and I'm going to try not to be overly redundant with what Anne showed you. So my goal here is just to kind of explain some of the background on network neuroscience as well as how we've been applying it. So feel free to stop me, ask questions. I don't have a particular agenda to get to. So yeah, so definitely stop me and ask questions. All right. So I just want to, can you all see my arrow on the screen?
We can.
Okay, good. I can't gesture with my hands quite the same. So I just want to shout out my collaborator, Josh, is a fourth-year graduate student, PhD student here at IU with Olaf, and I stole some of his slides to give this talk, so I wanted to give credit to that. And just to give some background, because I'm not sure, I think some people have familiarity with connectivity research, but just wanted to put us on the same page. So network neuroscience is a subfield of both network science and neuroscience that aims to characterize brain structure or function as a network. So really what it's focused on is relational data. And you know, the reason for doing this, for looking at relational data, is that the brain's a complex system. So it's rare that, you know, one region really acts alone. And then you can also study the brain at multiple different spatial and temporal scales. We're constrained by fMRI or MRI, which is what we use in our lab. But it's worth shouting out that what's really beneficial about a networks approach is that you can basically have the same type of data that you work with across scale and compare across scale. And so one example here from network science in general is flight paths. So airports are a network. So you can't really understand flight paths by looking at one airport, but you can look at how different airports here on this figure relate to each other, for instance, by the density of flights between those airports. And this is an example Olaf uses that I've adapted to corn mazes because it's fall. But you can't really understand kind of the broader properties of the system of a corn maze or how to get out of a corn maze by studying just one cob of corn. That is not to say that it's not valuable to study individual brain regions and what kind of cognitive approaches or cognitive processes are associated with individual brain regions. It's just a different type of understanding that we're getting from networks data that has its own limitations. So like I said, you can study the brain across multiple scales, not limited to fMRI, and these scales can be spatial. So along this red dimension here, you can study things. We have people here at IU who study, you know, population or ensemble recordings in animals. In MRI, which is what our lab uses, we're really looking at kind of the regional brain activation. And then you can look across topologies, so you can be interested in specific brain regions, the connections between specific pairs of brain regions, all the way up to kind of more global measures at the whole brain level. And you can also look across time. So in our case, we're interested in development, so how the brain changes as you go from adulthood to older adulthood, which I'm going to talk a little bit more about. But what I like is essentially you can kind of use a networks approach to characterize across a lot of different scales and dimensions. And minimally, a network needs nodes and links, which I'm going to call edges. So nodes are what comprises the network, and edges are the relationship between those nodes. And what comprises a node and what comprises an edge varies based on your methodology, your question, etc. So for instance here, I'm going to focus on regional networks within the brain using fMRI, but folks certainly, especially in social psychology, social neuroscience, have looked at social networks where people are the nodes and their relationships are the edges. And so I'm going to start by talking broadly about network construction, again, just to kind of put us on the same page about how do I get the data and then what we can do with that data. But feel free to stop me. So up at the top here, I wanted to use this figure so often, so I'm glad I get to talk about it. So up at the top here, we have a brain where you can get kind of the anatomical or structural aspects of the brain, as well as functional aspects measured by BOLD. So for instance, you can chop up the brain in a bunch of different ways, drop some pins in different locations and measure the signal across the time series. So across, for instance, a run. And I'm gonna start by talking about resting state. So resting state is, for instance, you put somebody in the scanner for 15 minutes, you ask them to stay awake, keep their eyes open, maybe fixate on a cross, but not, you don't give them a task to do, or you don't direct their thoughts. And so it's a way of measuring kind of unconstrained thought or undirected thought. And so using this data, you can create, I'm going to focus on functional brain networks, since structural brain networks come with all sorts of different assumptions that I'm less familiar with. So functional brain networks, essentially what you're doing is you're assessing the relationship in the time series data between every pair of brain regions that you're looking at. And so you can create a network that covers the whole brain. So every brain region with every other brain region. And then finally, you can, once you have that network created, you can do second level, group level analyses on these, which include graph theory. Graph theory is a type of mathematics used to describe networks that is sometimes conflated with network neuroscience as a whole, but it's not quite the same thing. So talking about defining our nodes. So what are our brain regions? Folks, this is an area of a lot of interest and really rich conversation right now. Generally speaking, a lot of the ways that people have derived networks have converged on a general description that looks like this. So this is from Yao's group, the 2011 paper. This is their 7 networks way of seeing what brain regions hang together using resting state functional connectivity from a large group of individuals. And so there are a variety of networks that they tend to identify, including, I'm going to focus a lot on this default mode network, which are the red regions here that include MPFC, PCC, as well as temporal parietal junction and temporal poles. So those are my brain regions, but then really I want to know, how are these brain regions communicating, relating to each other, that sort of thing. And most commonly, people use Pearson's correlations in the time series. So that's what they're talking about when they're talking about functional connectivity. There are other ways, but I'm going to focus on that because that's what I do. So for instance here, the orange line, so on the x-axis we have time, and on the y-axis we have % signal change. And we have 3 different brain regions represented. So we have MPFC in orange, PCC in yellow, and IPS in blue. And what you can hopefully see is that, generally speaking, the time series are pretty highly correlated between PCC and MPFC, and they're actually negatively correlated with IPS. You can also have no correlation, so no relationship in the activity, but that's the measure. So the strength of that correlation is how we're defining the relationship between brain regions, so the correlated activity over time. So now that we have our nodes and our edges, we can create a network. So we can get the data that we use at the first level. And so I'm going to take an example here. Let's say I drop a couple pins in the brain in these 5 regions. And I'm showing you, this is a surface of the brain, but typically you'd segment out the gray matter and define it similar to this. And then you construct a network. So really you're creating a matrix of values that reflect the relationships between these brain regions. So for instance, the correlation in the time series between blue and orange might be 0.3 if you're thinking about -1 to 1. And it's symmetric across the diagonal here. So the diagonal is the brain region with itself. And it's symmetric because it's not directed. So we're not making claims that orange is influencing blue or blue's influencing orange. It's just that they have some sort of relationship. There are approaches to get at directed networks, but that's kind of outside of the scope of what I'm going to talk about today. So just note that these are symmetric across the diagonal. So you create your matrix and you fill it in, essentially, until you get something that looks like this. So this is the data that I actually work with. I get one of these from every subject for every scan that they do, essentially. And then I'll note that along the diagonal, the correlation is 1. So we tend to kick those out because, they're not informative. So this is the data that we work with, and then how do we kind of describe the network using this data? Well, there are a variety of approaches. These are some of the graph theoretic approaches. I realize the resolution is not great, and I apologize about that. So over here on my left, I think you're right, we're looking at properties of weighted networks. So weight meaning that it's not just you're connected or you're not, but it's how connected, which is what Pearson's correlation gets you. Where each dot is a brain region, each line represents the strength of the connection. And there are a variety of aspects of networks that you can look at. I'm going to focus on modularity, so the actual structure of the network, because one of the most robust findings in the aging literature is that modularity changes with age. And what I mean by modularity is, for instance, this is how we want to kind of derive networks. So we want to know what brain regions hang together and what brain regions don't. And at rest, people have found that the brain has a property called small worldness. So brain regions that work together want to hang out together and don't want to hang out with other groups of brain regions. So here we get these distinct, non-overlapping groups of brain regions, and you could kind of zoom in and zoom out and get different groups, and that's a whole field. But this generally tells you about the structure of your data, where really there aren't a lot of connections between these except for maybe through one region. All right. So then what does the data look like in reality? So this is an example of a resting state functional connectivity data from a sample of younger adults that we have. And I've organized the data where the X and Y are every brain region by every brain region. Here we have 126, which is a relatively low resolution parcellation. And it's organized by the 7 YAO networks and then a subcortical network. And I'm showing you Pearson's R correlations because they're just nicer for visualization, but we do our analyses on Fisher-Z transform data. And what you can see is along the diagonal, we're talking about regions within the same network, you see a stronger density of these high positive correlations. Whereas when you're talking about between network connections at rest, like for instance, between ventral tension and DMN, oh, sorry. Okay. So for instance, here between ventral tension and DMN, you're seeing negative or weak correlations, no correlations, that sort of thing. So you see, this is the structure to the data, that when people are talking about a network organization or a functional architecture, this is what they mean. And there's heterogeneity, so visual is pretty tightly coupled with itself. DMN, you can see that there's some subblocks emerging, and that's pretty typical because the DMN here is quite large. I think it's 26 regions. So there's work in aging that suggests that certain subsystems of the DMN are more impacted by aging than others. So in general, it's not surprising to see kind of sub-networks emerge. To show you it's not just our data that we find this in, I'm showing you 56 minutes of rest from Michael Cole's 2014 paper. He used a different parcellation, so he has 264 regions, more networks that he's looking at, but essentially you see kind of a similar pattern that along the diagonal, you see high positive correlations within network and weak or negative correlations between networks at rest. All right, so our lab does a lot of work in aging, and so I'm going to talk a little bit about that. So I told you we're interested in network structure, modularity is what it's called. So here I'm showing you young and older adults, young over here, older adults over here. And this is a different way of depicting the structure of the networks. So for instance, younger adults, this is default mode, I think, in red, and older adults. And then what you can generally see from this is younger adults networks like to be separate and more pulled apart, whereas older adults networks kind of don't have that same quality of separation or segregation. This is across the lifespan. So when we start looking at middle adults, middle early and middle late is what they're called here, you see kind of that progression of greater integration among older adults and the trajectory at which that takes across different types of networks. So this is what I'm talking about when I say that older adults are kind of losing that network structure. And this is all resting state data. So now, if everybody's on the same page about that, I'm going to just talk a little bit more about our kind of how we do things specifically in the lab, and hope that, you know, I'm giving you some tools that could be useful. So our lab got into this about 4 years ago. When I joined the lab, I really wanted to do network science and apply it to some of the social neuroscience questions that we had. And so I went to Olaf and Josh and I said, hey, I need your pipeline. I need to know what is the way to do this, the agreed upon, nobody's gonna, you know, kick me way of doing this. And to be honest, it's not that straightforward as with most questions. So what I'm showing you here is just a snippet of what we've landed on is what works for us, what answers our questions in the best way, in our opinion. But there are a lot of different ways of implementing these analyses. So for preprocessing and network construction, we like to use the functional connectivity toolbox, and the website and associated citation are listed down here. I'm happy to send these slides after the talk if it's useful for folks. I'm not going to get into Con because they have really excellent tutorials, videos. They have a ton of materials on how to use their toolbox, and it's really well developed in my opinion. As somebody who didn't have programming experience coming into grad school and then not only had to learn how to use, you know, MATLAB, SPM for GLM analyses, but also then for networks, I found it quite user-friendly. So we use MATLAB and SPM for GLM analyses, so it was nice that this kind of wraps around those. And then an issue with resting state, especially when you're looking at cohort differences, for instance, older adults versus younger adults, is you need to be very careful about your denoising and your motion artifacts, some physiological noise associated with heartbeat, respiration, because you don't really have the same sort of subtraction process that you do during task activation analyses. And this is just one approach. It's pretty commonly used, but it's not the only one. So CompCore is a PCA-based approach. And to estimate noise as it varies across the brain, in contrast to global signal regression. You may have heard about this, like, for instance, via Twitter recently. So global signal is kind of a key pre-processing issue that people tend to do it with and without, just to make sure that the results aren't based on how they pre-process the data. But global signal averages the signal across the brain and then removes it. But motion is pretty important in older adults, and what we find is that older adults move more, and we have to account for that in the quantity of data that we collect to make sure that we still can attain reliable functional connectivity estimates, even if older adults are moving more. Okay, so I mentioned a lot of this. They have a really nice GUI, and they also have good scripting, so you can script if you want and totally work within that. And you can also use the GUI. I like to use kind of a combination just to make sure that things are loading properly in the way that I think they are with the scripts. And another nice thing is that you can really customize what you want to do. So you aren't beholden to a particular parcellation. So you don't have to use one, you can use multiple, you can use custom ones, and you can customize every aspect of the pipeline. And the caveat to all this is I've mostly used this for preprocessing and network construction. I haven't used it for second level group analyses or visualizations, though there's great documentation on that. This is just a sample from Con's website of all the things that you can do in Con, of which we kind of focus on one thing. And just to give you a sense of what this pipeline looks like, so you set up, just like anything, you give it your data, you tell it about the experiment design. So I'm going to talk a little bit about task evoke connectivity later. So here for resting state, you just say everything from 0 to infinity in the scan is one thing. That's nice about resting state, is that you don't have to add a lot of complexity in your design. But when you have task designs, you need to specify that to a greater extent. And then you can specify what regions you're talking about. So let's say you have a theoretically theory-based reason for focusing on MPFC projection, so how the MPFC is talking to other regions. You don't need to do every region with every region. You can just look at how the MPFC is talking to other brain regions. So you pre-process your data in all the ways that, you know, you do. And then you do kind of some further denoising, removing of physiological artifacts, subject motion, and they give you really detailed quality analysis information at every step. You do your first levels and then your second levels. Looks pretty basic. Again, I'm kind of working mostly within the Roy domain. And then when we're talking about first level or second level analyses and visualizations, we use a mix of custom code and the Brain Connectivity Toolbox. The Brain Connectivity Toolbox is you know, publicly available. It's MATLAB-based scripts that are fairly easy to use. And they're meant to give researchers, once you have those matrices that I showed you earlier, you can use this to run all the graph theory and all the different network metrics that you want. Which includes some of these things listed here, including modularity. They've recently expanded, I think just this year, into giving you some tools for network construction, for community detection, so figuring out what are the brain regions that hang together, creating null models, which I'll talk a little bit about, which are useful for significance testing, and then visualization. But really, the meat of this toolbox is focused on giving you those metrics that characterize the network. And it is a plug because Olaf helped develop this and his lab maintains it, which is part of the reason why we use it. But there are other connectivity toolboxes out there. So now I'm going to get into our data and hopefully not rehash what Anne talked to you all about, but kind of give you some more methodological information and some considerations for how we kind of had to move within this space, especially considering that we're looking at cohort differences, which I found is one of the things that reviewers tend to really ask questions about in our data. Is not so much, you know, did you slice time correct or not, but how did you account for brain size or volume differences between old and younger adults? Is it appropriate to really kind of register them to the same template? These sorts of things, or use the same parcellation. And, you know, there's answers for that. If we want to talk about that. So in our data, we have 40 young and 35 older adults. And we would love to have many more people. There are definitely data sets out there that are publicly available, like Human Connectome Project. I don't know if that's publicly available, but there's the Cambridge Aging Project. And also, if you're not interested in aging, there are other data sets with healthy adults, as well as everything you can think of, I'm sure. So this is a relatively small sample compared to some of those data sets, although it's kind of typical in the field now. So I'll caveat that, you know, a lot of times when looking at brain behavior correlations, the sample size, it's okay, but it's not perfect. And then we collected data across 2 sessions. In the first session, we got, the reason why we collected our own data versus using these others is we wanted to answer questions about specific social cognitive behaviors that were of interest to us, but not necessarily captured in these larger data sets. So we did some behavioral testing in the first session and MRI testing in the second. We did 15 minutes of resting state, and really, I think the field has really shifted towards 15 minutes being like the minimum that you want to collect. Resting state, again, to get kind of reliable estimates to, if you're trying to identify like who's young, who's old, and do brain behavior correlations. More data is better. Of course, at some point, there's no additional benefit, but there's a lot of great work on kind of this topic of how much data to collect. So we were fortunate to really to collect 15 minutes and be able to do that. And then we also collected 3 task scans related to social cognitive abilities that differ with age. I'm going to focus a lot on the theory of mind task that we had because it had the greatest quantity of data, but also has a performance aspect based on accuracy where the other 2 don't. So it's a little bit cleaner in its interpretation for these analyses. And just briefly, the theory of mind tasks, essentially participants read a story and then were presented with a statement about that story that they determined was true or false. And we had trials where participants had to make inferences about what other people know, so regarding theory of mind, and then control trials about objects. So in our first study, we were interested in establishing whether resting state connectivity within this default mode network predicted theory of mind performance on this task scan. And the reason why we were interested in rest is that there are really robust differences, age differences at rest. So potentially it's what's happening at baseline rather than how older adults actively engage in tasks that's underlying performance deficits in older adults. And we focused on the default mode network because the neural correlates of theory of mind, which I'm showing you here, show extensive overlap with the default mode network. So there have been meta-analytic studies confirming kind of the convergence of these locations, so defining our nodes. And then we were interested in whether or not age differences at rest related or explain the age differences in performance. And what's cool about this study that I think is pretty important to think about when you're doing networks analyses is, are we talking about more global or more local effects? So here we start at the level of the DMN, which I said is pretty big. It's 26 regions. It covers a lot of brain. We focused on a theory of mind network, which I'll talk about how we defined, but that's nested within the DMN. And then we focused on particular edges or the singular connections between different pairs of regions within this theory of mind network. So we kind of tried to scope in to see where do age differences emerge, but also where do we predict behavior most robustly? So what we find is that age differences emerge across scale. So we see age differences at the level of DMN. So here I'm showing you older adults in red, younger adults in blue. And Q is a measure of modularity. So how strongly is the network kind of hanging with itself and not hanging with other networks. And this is at rest, so it really should be hanging with itself. So we see that younger adults have greater modularity of the DMN, as well as this theory of mind network. Of course, there's a fair amount of overlap, and that's what we see with behavior too, is that these are high functioning older adults. So we're not expecting like huge differences in brain activity or behavior. The key finding here was we defined a theory of mind network using the task scan. So we took the peaks of the activations, which I'm showing you here on the surface, from the theory of mind localizer, which identified these kind of commonly identified regions associated with theory of mind within our subjects, and then found where the peaks fell within the parcellation. I identified, these are the outlines I'm showing you here. Selected where the peaks fell, and then characterized the resting state connectivity between these 8 regions. So it was a way of narrowing in our network to say, is it kind of the property of the network, or is it something about the relations between specific brain regions at rest that may be predicting behavior? So this is a network that we created, visualized in a different way, where each dot around the edge is these 8 brain regions. And then each line represents the age difference between the brain, in the connectivity between brain regions. So blue here is where the connectivity was stronger in younger adults, and red was stronger in older adults. And what you can see is largely things are stronger in younger adults, which is consistent with past work. And after we did some corrections for multiple comparisons, 2 edges emerged as having significant or very robust age differences, and it was between right TPJ here and right temporal pole, and then MPFC here and the right SFG up here. And so then these are where age differences were most pronounced, but which predicts which or both predict behavior was a question for us. And we started to zoom in on the edges because we didn't find that the property of the network, the modularity of the DMN, or of this 8-region network predicted behavior. So it didn't relate to behavior for old or younger adults. So Anne probably talked about this, so I'm going to go through it kind of quick. We did just basic linear regression on the connectivity of the edge, so the actual just R value. And what we found was within this particular edge between right TPJ and temporal pole, the stronger connectivity related to better theory of mind performance. So this is a difference score where you're not performing better on the theory of mind trials than the control trials. And that's what we would expect based on the literature. So younger adults are really driving this. So the relationship is stronger among younger adults. So that's how we got to the conclusions of our first study. Really, we're not doing anything overly fancy, but just kind of characterizing where do age differences emerge and where do they predict behavior in resting state. Some more recent work that just went under review is now looking at what's the relationship between rest and task states among older and younger adults. The reason why this is important is oftentimes when we're thinking about task fMRI, we're comparing two conditions with itself, and we're looking at very local effects. And so we're not really sure what's necessarily happening at the wider scale or the more global scale of brain connectivity in this case. So Michael Cole's group has a paper from 2014 where they determined based on HCP data as well as data from in-house that rest and task are highly correlated, the connectivity. So they're very similar, like eyeballing this plot, it makes sense that there's a correlation of 0.9 between them. Our question in the context of aging, knowing how aging disrupts multiple systems across the brain, it should lower the correlation. So the extent to which older adults' brains change between rest and task or differ between rest and task should be greater, reflecting a lower correlation. The way that we tested this was we took these connectivity matrices. So right now I'm showing you younger adults at rest, which I showed you before, and younger adults during the task. This was the theory of mind task, but we just did it across the whole run, so across both trial types. And essentially what we did, I'll show you the older adult data too. So this is the whole brain, just kind of getting the bird's eye view. So older adults at, younger adults at task, what you can see is, generally speaking, hopefully you can see this, generally speaking, you preserve some of that network structure, so the strong within network connectivity during task, but you see more between network interactions. So you see stronger connectivity happening between networks, for instance, the DMN and ventral attention network. So there's greater change happening between networks. Older adults start with weaker connectivity at rest, which has been widely established. So here I'm showing you that the strength of the correlations within network are weaker among older adults. There's also some stronger between network connections, reflecting the extent that their networks are no longer so segregated, but more integrated. So putting the younger adults task up, what we thought was really striking was the older adults at task here. What's happening is that they're not just showing the same pattern at rest, but in fact, they're weaker within network, and then they even start weaker than younger adults, but also they see greater between network connectivity. So I think I'm about to show you this in a slide or two, but essentially we're seeing a greater amount of change or difference between rest and task states in older adults. And if you look at the DMN, for instance, just kind of zooming in on it, you can see how it's roughly preserved here. Older adults start weaker, but that structure seems to fall out largely. The visual network is pretty tightly coupled no matter what you do. So to look at the correlation between states, what we do is adapted from Michael Cole's group, which is essentially we take all the non-redundant connections because the matrix is symmetric. We put them into vectors. So this is just random numbers, not real data. This is real data, that's not. With rest and task, and then we calculate the correlation for every subject between rest and task, the same edges at rest and at task across the whole brain. So I'm talking right now about very global effects in the brain. And then when we do the group comparison, we find that younger adults have a higher correlation in the connectivity between rest and task, whereas older adults have a lower and more variable correlation. What was the point I wanted to make about this? Yeah, so this is quite striking in our mind. But it makes sense based on the patterns that I was showing you at the bird's eye level, that there seems to be kind of this falling out of the network structure. And I'll mention that the way that we do group comparisons here when we do cohort testing is we do permutation tests for significance. So what this looks like in brief is we have our group of young and older adults, and then we shuffle them, because if we think that age is the distinction that matters, that's really driving these effects, then by shuffling the group assignments but preserving the group sizes, and then recalculating the difference, the t-value in the age difference and the correlation, we should create a null distribution if we shuffle this over and over, and then we can test the significance, the extent that our data is unlikely compared to chance. Of course, there are more conservative null models. So it's nice to create a null model based on, your real data or valid experimental data. This is a whole debate as well, but you can add in other factors that you think co-vary with age. So just shuffling this may capture other things. So you can try and control for those factors to really say that it's age driving this and not other things. Right, so I showed you this. So why does this occur? So why do we see a lower correlation? Well, I was just telling you, it seems like that network structure is falling out. So we can actually test this using some of these network neuroscience tools to describe the organization of brain regions into networks, functional brain networks in this case. So what we did was we did community detection, which is a way of kind of finding out what is the best description of the organization of these brain regions into networks. And we varied along gamma. Gamma is a resolution parameter, so there's no right answer about how much to zoom in. So if you're taking a picture of my face, you could get right here, or you could be pretty zoomed out. Both are valid, but it's probably like a mid-range. That's the best picture of my face. Hopefully I'm the appropriate amount of away from the screen for this to take real effect. So we vary gamma as a robustness check, but it's not just due to the particular free parameter that we chose. So to give you a toy example here with the networks, we have 6 brain regions in each of these blocks. And let's say younger adults at rest, they fall out. The best description of them is 2 networks represented by the color. Younger adults at task, because we're seeing more between network interactions, which we would expect, because at rest, we expect them to kind of hang out separate, but they should interact in order to flexibly kind of produce the behaviors that we're interested in. So we see more between network interactions. So maybe there emerges a 3rd network. So there's a degree change of 1. In older adults, what we see in our data is that older adults start with maybe more networks or a less parsimonious description of the network. But also during tasks, they see more change between rest and task. So two of these blocks flip assignments. And so you really don't get a very clear parsimonious description. So that's what we tested in our data. And we did this across both the theory of mind task as well as our face rating task. So we tried to show it across that it's not something specific to task at this point, that we're really just showing what's happening when older adults are engaged in rest and when you add sort of exogenous demands to it. So here in red, we see younger adults at rest, where X is our resolution parameter. So when you're really close up on the brain, or when you're really far away, you're only really distinguishing hemispheres. So right hemisphere hangs together, left hemisphere hangs together. At really high values of gamma, you're getting individual brain regions do everything differently, which we know is not really how the brain works. So looking kind of across the span here for robustness, we find that mostly younger adults have fewer networks at rest and a less parsimonious description at task. That's this green line. Older adults start higher at rest and then also see an increase at task. And once you're getting up to like 50 brain region or 50 subnetworks, we have 128 brain regions. That's 2 or 3 brain regions per network. So it's not very parsimonious. And that's all this figure is trying to show you. And the benefit of what we're doing here, comparing rest and task within groups, so rest versus task within younger adults and rest versus task within older adults, is we're getting around some of the issues that you may find with older adults have generally lower connectivity or start at a different baseline. So by waiting to do the age differences once we compare within state, we get around some of these issues with cohort differences, or considerations, I should say, not issues. The other thing we did here was we measured how similar, how consistent the network structure is. And why this is important is what Cole and other people have found pretty nicely is that, generally speaking, the structure, the architecture of functional connectivity within the brain is relatively consistent regardless of task state or rest. And this makes sense from an efficiency perspective, because you should have fewer connections that are really being modulated when you engage in different types of tasks, and preserving kind of the larger structure. So what we should see in younger adults, because they're more correlated, is that you see greater consistency between states. And that's what we find. So this is a measure of how inconsistent the network structure is. So this is talking about the structure of the data, not the connectivity itself, but the ways in which these networks are organized. And so younger adults in red here have more consistent network structures between rest and task states than do older adults across a range of gamma. So we had some predictions. So this is all looking at a very global scale at the whole brain level. Because the DMN has often been implicated in aging, we decided to kind of zoom in and say, are there particular networks or blocks between networks that show a greater degree of change between rest and task? And I should caveat, we're talking about change at a very temporally broad level. So we're not talking about like moment to moment fluctuations. And so we expected that the magnitude of difference or change between states would be greater in older adults in the DMN, and it's because the correlation is lower. The way we tested this was actually to go back to the edge level and look for age differences and the extent of difference between states at the edge level. So we have our rest and task data. These are correlations. We take the difference between rest and task, reflecting the extent of change. And then we simply take the absolute difference. So regardless of whether task is increasing or task is weaker connectivity relative to rest, we're ignoring at this point, but I'll come back to it. And then if you do this across the whole brain, these are the difference matrices. So the difference in correlation values between rest and task for young and for old. So showing in blue here, this is task minus rest. So things within network are stronger at rest, and things between networks are stronger at task. And this is more pronounced because the colors are darker for older adults. So what we use to look at blockwise differences in specific networks is network contingency analysis, which looks at the counts of the edges rather than like averaging across all the edges, because it just gives you kind of a better resolution regardless of how you define your blocks. You can do this type of analysis, versus if you average in different ways, you kind of lose some of that specificity. So this is adapted from Sripada's paper in human brain mapping. They use a slightly different approach, but it's very similar. So we have our connectomes or connectivity matrices. We do a t-test at every edge between older and younger adults, not significance testing, just getting the t-value. So where positive ones mean it's greater in one group than the other. Then we threshold across a range, because the threshold is really, it's like you could use significance testing and use like an FDR correction or whatnot. We like thresholding the t-values because it's more direct to the data. And so you get each of these dots reflects a value that survived the threshold. And then they fall within these blocks of the networks, which looks like what I showed you before. And then we simply count these based on chance, based on the permutation testing. And so we highlight the blocks that show age differences in the magnitude of change between states. And what we found was that mostly the magnitude is greater, or completely in this case, the magnitude is greater in older adults, where we see these three within network blocks showing a greater change between rest and task, a greater difference in older adults, as well as one network between one block between networks. And so this figure is a bit misleading because it's not saying what's higher in rest or what's higher in task. So then we did, to get converging evidence, we did the average connectivity, having identified these blocks based on the network contingency analysis. And so what I'm showing you here, average FC is average functional connectivity, higher values is stronger. Younger adults, for instance, in the DMN here, they decrease, but not that much, whereas older adults, the magnitude of change is greater, and it's driven by the fact that at task, that within network connectivity just seems to wash out, which checks out when you look at the between network connectivity increases to a greater extent in older adults and younger adults. So that kind of gets you through the analysis all the way from data collection to preprocessing to network construction to some of the analyses that we've done so far. Some ongoing work I'll touch on briefly is the task evoked connectivity that we were looking at before really only answers the question of what happens when older adults do a task. So when they're doing something versus not doing something. And oftentimes in like social neuroscience, we're interested in what's happening when you're thinking about yourself versus somebody else, when you're engaging in theory of mind versus a different task condition, and then also building in rest to that question. And so that's where we're going next with this work. So we're really looking at task-specific or condition-specific connectivity. For instance, this is the behavioral performance on the theory of mind task, where we have two conditions. So we find that older adults are really impaired compared to younger adults, although everybody's performing really well in the theory of mind trials and not the control trials. And so because we have this behavioral deficit in one condition, not the other, we can look at the corresponding connectivity to see if we can say something about, is it rest, is it task connectivity in general, or is it like how older adults do any task, or is it how older adults are doing this task? That's them relating to their behavioral changes and at multiple spatial scales. Yeah, and then my dissertation committee really wants me then to answer the question of, is it activations or is it connectivity and connectivity between all these states? So that's kind of where I'm going next. Anne said folks had questions about task connectivity, so I'm going to touch on that a bit. Essentially, task connectivity is a bit tricky, especially if you have an event-related design, so block designs are probably better. I've seen folks who have like the whole run is one type of task and a separate run is another type of task, or you just have longer block periods. So typically I've seen, and ours is 18 to 24 second blocks. I've seen that's pretty typical in what I've seen like with Human Connectome project. And then what you want to do is regress out the task events. So both the onset as well as the duration of the task events in the condition you aren't interested in to get what's left over in the co-fluctuations. So not the co-activations, but how they're moving together in time. And I like this paper from Cole, this really recent paper, and how he and his group described this in terms of, they found that activations spuriously inflate correlation estimates. So your inference here is maybe they're not talking to each other, but the correlation is higher just because of the task timing. So really all you can conclude if you don't regress out task events is they're either interacting or they're both just active during task. Whereas if you do a task event regression, then you can say that they're likely interacting during the task or they aren't. But of course, this is still kind of the Wild West in what I found. So people do things in different ways, and really you just kind of got to dig into it a bit. And the quantitative data is really helpful. So like I said, it's the Wild West. Resting state, I mean, people still do 5-minute scans, 8-minute scans, but really the field is moving towards more data is better, especially when you're doing cohort studies or trying to identify biomarkers. So with task scans, we find that our effects are condition or state dependent. So we find greater age differences at task than rest. So I think that, while we're looking at really like 3 to 8 minutes of task in the studies that I've seen, and these 20 to 25-second blocks, I think collecting more, having longer durations, being able to better pull apart, like if you have 2-second events, like we have a face task where we just show faces, 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 I wouldn't do connectivity with that. So I think just keeping that in mind if you're really interested in task-evoked connectivity and splitting out conditions, that's the quantity of data is really important, especially since you lose a fair amount of that with motion. And so just some take-home points. Based on my experience, again, so build in robustness checks. So really I found that there's no one answer. So if you think, should I use this parcellation or that parcellation? You should probably try both just to make sure your results aren't due to how you defined your brain regions, your nodes, or based on some preprocessing choices. Null models and how you create them are really important for statistical testing. Try to replicate your findings. So we did this across multiple tasks. We tried to replicate what we could across the tasks that we have in our sample, but also testing out of sample or doing leave one out cross validation. And then just cohort difference in general, because you're testing older adults and younger adults at 2 different states, you're really testing interaction predictions. So there are a whole bunch of different things that you have to account for in terms of cohorts and what could be different apart from just what you're interested in. And then finally, Olaf has really hammered this into my head. Figures need to be clear. So the cool thing about networks is they have beautiful figures. You can add all sorts of colors and dimensions and information in your figures, but oftentimes they're overwhelming. So you just want to make sure your figures are really tight, really clear in what they're communicating. The colors are clear. You don't have too many. This seems obvious, but I think I've really had to think through how I present my data a lot more carefully working with this. And none of this work happened in isolation. Josh walked me through quite a bit. We spent about a month just talking about global signal regression. So I don't get any of this done without my team. And then shameless plug, I'm looking for a postdoc. So if you all like what I do and want to share that information, feel free to. And that's what I got.
Thank you, Colleen. So we have just a few minutes for questions.
I can't see if anybody's raising their hand, so
I'll raise
go ahead.
Yeah. Introduce yourself as well.
Hi, Colleen. My name is Heather, and I work in a connectivity lab as well. We also study different cohorts, except we go the opposite direction with development. We study neonates instead.
Yeah.
So you kind of touched on this a little bit in your presentation. I want to probe you a little bit more about how you account for differences in brain size. Can you correctly, transfer things from different sizes? Are parcellations accurate? Things like that. How do you address that?
Yeah, that's a really great question. What I've seen and what we've tried is one big thing with when you're parceling out the brain and aging is that older adults have shrinkage. So the actual amount of area you're covering may be different, which gets at the parcellation issue. So we try and covary out with our connectivity, gray matter volume. What else have we tried? We tried other demographic variables kind of to see how much it changes the results. To the point about parcellations, I think there's really cool work being done at UT Dallas and Miami where they're looking at how to create parcellations based on different lifespan stages that account for some of these issues and how maybe the definition of a PCC, for instance, may differ across the lifespan and how to capture that while still being able to compare. Because if you use different parcellations for older adults and for younger adults, can you make the same comparisons? And so folks are trying to define networks in such a way that you can retain being able to look across cohorts. Does that help?
Yeah, it does. Thank you.
I think we're all but out of time, unfortunately. Everyone, please join me again in thanking Colleen for her wonderful talk. So just a few quick announcements before you go. If you haven't already, please sign in to the sign-in sheet. It's how we maintain food. Also, look out for some emails about some future upcoming talks that we have in the works. Thanks, everyone.
You're gonna take more food before you leave?
Oh yeah, and Amanda has a quick announcement.
I wanna go.